Dear students, we continue our study of the course of special histology. Today we will consider the features of microscopic structure and functions of the main part of the digestive system, the alimentary canal. The digestive system plays an important role in our body, performing a number of vital functions, foremost of which are mechanical and chemical food processing, absorption of monomers and excretion of metabolic waste products. The organs of the digestive system also produce hormones and are involved in breathing, speech and immune protection. The digestive system consists of the digestive tract, or alimentary canal, and the large glands associated with it and located outside the digestive tube. These are salivary glands, liver, and pancreas. The digestive system consists of three sections: anterior, oral cavity; pharynx, and esophagus; middle, stomach, intestines, liver and pancreas; and posterior, caudal part of the rectum. The wall of the digestive tube of different sections is built according to a single plan. It is formed by four membranes or tunics: mucosa, submucosa, muscular layer, and serosa or adventitia. The mucous membrane consists of three layers: epithelium, the lamina propria, and muscularis mucosa. The epithelium and the lamina propria are obligatory components of the mucous membrane and are well expressed in all sections. The muscular mucosa in different parts is expressed to varying degrees or may be absent. The epithelium in the anterior and posterior sections is stratified squamous non-keratinized. In the external border zones it is keratinized, and in the middle section it is simple columnar, which lies on the basement membrane. The epithelial cells produce mucus for protection from chemical and mechanical damage. The lamina propria is a loose connective tissue with a dense network of blood and lymphatic vessels and lymphoid nodules. In the stomach it may contain glands. Adjacent to the lamina propria is the muscularis mucosa, consisting of from 1 to 3 layers of smooth muscle cells. The submucosa is also loose connective tissue with the same components, but contains more fibrous structures. It provides mobility of the mucosa and the formation of folds. It also contains glands in esophagus and duodenum. In some areas such as gums, hard palate, dorsal and lateral sides of the tongue, the submucosa is absent. The muscular layer is formed by muscle tissue. Its contraction provides mixing and motion of the contents of the digestive tract in the caudal direction. In the anterior and posterior sections it is represented by skeletal muscle tissue, and in the middle, smooth. It includes from 2 to 3 layers, separated by the connective tissue, or can be completely absent in gums and hard palate. The serosa of the digestive tract consists of loose connective tissue and mesothelium, simple squamous epithelium. The epithelium secretes and absorbs serous fluid, making the surface of the internal organs smooth, moist and slippery, which facilitates the movement of closely packed organs and their parts in the abdominal cavity. In the esophagus above the diaphragm and larynx the serosa consists of loose connective tissue and it is called adventitia. This tunica is absent in tongue and lips. The anterior section of the digestive system includes the oral cavity, pharynx, and esophagus. Derivatives of the oral cavity include lips, cheeks, gums, palate, tongue, tonsils, salivary glands, teeth. This section ensures the capture of food, its mechanical processing and movement to the middle section, and also provides respiratory and speech functions. The anterior section contains taste buds and structures that provide immune protection. Lips perform tactile and mechanical functions. These are skin muscular folds, which have the outer and inner surfaces. The outer surface or external region of the lip is covered with skin. The inner surface or internal region is an oral mucous membrane lined with stratified squamous non-keratinized epithelium. The underlining lamina propria of the mucosa passes into the submucosa, where the secretory portions of the salivary glands lie. Their ducts open onto the surface of the mucosa. The transitional region constitutes the free vermilion border of the lip. This zone has only a thin, lightly keratinized epidermal covering. The dense network of small blood vessels is responsible for the red color of the lips. This part is highly sensitive due to its rich sensory innervation. Teeth provide mechanical breaking down of food. A tooth consists of a crown, neck and root. The crown projects above the gingiva. It is covered by enamel. The roots of the tooth project below the gingiva. They are covered by cementum and located in the socket of the bone jaw, lined with periosteum. With the help of the periodontium, circular ligament, the tooth root is firmly fixed in the bony socket. The neck is the intermediate part between the crown and the root and is covered only by the gingiva. Inside the crown is a tooth cavity, the pulp cavity. It is a fine fibrous loose connective tissue penetrated by blood vessels and nerves. The cavity of the tooth continues to the apex of the root, a root canal. Through a hole at the top of the root, blood vessels and nerves penetrate into the tooth. Enamel is the strongest tooth tissue, containing 3–4% of organic substances. The enamel is composed of enamel prisms glued together with the same durable substance. Ameloblasts are responsible for the production of enamel. They are absent in the formed tooth. Beneath the enamel is dentin, which makes up the bulk of the crown, neck and root of the tooth. It is a type of bone tissue and is characterized by significant strength. The main substance of dentin consists of bundles of collagen fibers and adhesives impregnated with calcium salts, and is penetrated by tubules. Nutrients pass through the dentinal tubules to the dentin. The dentin surface in the crown area is covered by enamel, and the root area is covered by cementum. Odontoblasts located in the surface layer of the pulp are responsible for the production and condition of dentin. They are pear-shaped, have a basally located nucleus and one long and thin process, which is located in the dentin tubule. Cementum is a type of coarse fibrous bone tissue. It is less durable than dentin and enamel. Bundles of collagen fibers penetrate from the periosteum into the cement. Cementocytes located in the thickness of the cementum are responsible for the production and condition of cementum. The tongue provides mechanical processing of the food. It is the most important organ of speech production and provides taste sensitivity. The mucous membrane of the tongue is covered with stratified squamous non-keratinized epithelium. Its mucosa forms 4 types of papillae: filiform, fungiform, foliate, and vowlate. Taste buds are located in the epithelium of the side walls of the papillae. The taste bud is made up of sensory cells, supporting cells, and basal cells. The plasmalemma of the apical layer of sensory cells forms microvilli with receptus. On the lateral surface of sensory cells there are endings of nerve fibers. The submucosa is present only under the mucosa of the lower surface of the tongue. The modified muscular layer of the tongue is formed by bundles of skeletal muscle tissue oriented in different directions, between which lie the terminal sections of tubuloacina salivary glands, producing mucus and serous mixed secretions. There are three pairs of major salivary glands: parotid, submandibular and sublingual. They produce saliva and their ducts open into the oral cavity. Saliva moistens and softens the food, cleans the oral cavity and controls the bacterial flora by lysozyme use, promotes the formation of a food bolus and its swallowing. Salivary enzymes perform the initial digestion of polysaccharides, nucleoproteins, and proteins. Biologically active substances in saliva stimulate epithelial growth, secretion of gastric glands, and other processes. The secretory cells of the salivary glands lie in one layer on the basement membrane, and the myoepithelial cells located outward contribute to the rapid removal of secretions. The cells of the serous sections have weakly basophilic cytoplasm and spherical nuclei. The cells of the mucous sections, mucocytes, are larger in size than serous cells, have flat nuclei, shifted to the basal side and are weakly stained using standard staining methods. The terminal portions of the salivary glands are serous, secreting mainly enzymes; mucus, secreting mucus; and mixed. In the mixed terminal portions, mucous cells occupy the central part, and serous cells, in the form of demilunes, are located on the periphery. The excretory duct system includes intralobular ducts, interlobular ducts, and the common excretory duct. Intralobular ducts are represented by intercalated and striated ducts. The striation of the intralobular ducts is due to the folds of the basal part of the plasmalemma. Mitochondria are located in them. At the apical pole of the excretory duct cells there are microvilli. According to the method of secretion, the salivary glands are classified as merocrine glands. The outside of the glands is covered by a connective tissue capsule, from which septa extend inward, dividing the glands into lobules. The parotid glands are compound-branched alveolar glands. Secretory portions are formed by serous and myoepithelial cells. The submandibular glands are branched tubuloacina mixed glands. They contain two types of secretory portions, serous acini and mixed tubuloacini, which contain mucous cells and serous demilunes. The sublingual glands are also branched tubuloacina mixed glands. They contain three types of secretory portions, serous, mixed, and mucus. Mixed and mucous acini are predominant. The esophagus performs the function of transporting food to the stomach. The mucous membrane of the esophagus forms longitudinal folds. It is covered with stratified squamous non-keratinized epithelium. The lamina muscularis of mucosa consists of a single layer of longitudinally oriented smooth muscle cells. The submucosa is well defined and contains the terminal portions of numerous complex tubuloacina, predominantly mucous glands. The muscular layer in the upper third of the esophagus is formed by striated muscle tissue. In the lower third it is smooth, and in the middle third it is mixed. The cervical part of the esophagus is covered with adventitia, and the thoracic and abdominal parts are covered with a serosa. The middle section of the digestive system includes the stomach, intestines, except the caudal part of the rectum, liver, and pancreas. In the middle section, chemical processing of food, absorption of nutrients into the blood and lymph, production of biologically active substances, excretion, and formation of feces occurs. The stomach produces enzymes involved in the digestion of mainly proteins, performs a mechanical function by transformation of food into a chyme, and produces a number of hormones and biologically active substances such as gastrin and histamine. The walls of the stomach consist of mucus, submucous, muscular and serous membranes. The mucous membrane forms folds, fields, and pits. The mucosa is covered with a simple columnar glandular epithelium. It produces mucus. In the lamina propria of the mucous membrane there is a large number of simple tubular glands, among which there are cardial gastric glands proper and pyloric glands, located in the corresponding areas. The glands open at the bottom of the gastric pits. The lamina muscularis of the gastric mucosa consists of 3 layers of smooth muscle cells. The submucosa is well developed. The muscular layer of the stomach consists of 3 layers of smooth muscle cells. The outside of the stomach is covered with a serosa. At the junction of the esophagus with the cardial part of the stomach there is a sharp transition from stratified squamous non-keratinized epithelium to simple columnar epithelium. The gastric pits in this area are about 30% of the depth of the lamina propria of the mucous membrane. The cardiac glands have wide, coiled tubular terminal portions. The cells secrete amyloid enzymes that break down amylin. There are also endocrine cells that produce gastrin. In the fundus of the stomach, the depth of the pits is about 20% of the thickness of the lamina propria of the mucous membrane. The fundic glands are elongated, weakly branched, and located very closely to each other. Gastric glands proper are the most powerful and numerous glands of the stomach. They contain 3 main types of cells: chief, parietal, and mucus. There are also cambial and various endocrine cells. Chief cells are the most numerous. They produce the inactive enzyme, pepsinogen. The parietal cells are much larger than the main cells, oval in shape, and are located on the surface of the main cells, wedged between them. They produce chlorides, from which hydrochloric acid is formed. In its presence, pepsinogen is converted into pepsin. Mucous neck cells are few in number. They are located at the neck of the gland and secrete mucus. Stem or cambial cells are low columnar cells, have high rate of mitosis. Some of them differentiate into surface epithelium, another in glandular epithelium. Different types of endocrine cells produce histamine, serotonin, melatonin, and other hormones, regulating gastric motor activity, gastric secretion, and blood supply of mucosa. In the pyloric part of the stomach, the depth of the pits is 2/3 of the thickness of the lamina propria of the mucosa. The pyloric glands have short branching terminal portions. They mainly secrete mucus. As in the cardiac glands, there are also endocrine cells that produce gastrin. The pyloric part of the stomach has the most powerful muscular layer. Here it forms the sphincter. In the small intestine, the chemical processing of all components of food masses, the process of absorption and the production of biologically active substances continue. With the help of peristalsis, the intestinal contents move in a caudal direction. The relief of the small intestine is characterized by the presence of folds, villi, and crypts. Villi are finger-shaped outgrowths of the lamina propria of the mucous membrane into the lumen. They consist of lost connective tissue, covered by simple columnar epithelium. Inside the villi there are blood and lymphatic vessels and individual smooth muscle cells located vertically. The presence of villi increases about 8 times the surface area of epithelial lining of the small intestine. The intestinal crypts or Liebig-Jones glands are tube-shaped recesses of the mucosa in the full depth of the lamina propria to the muscular lamina mucosa. The epithelium of the mucous membrane is simple columnar, bordered. The main cells of the epithelial layer are enterocytes. Bordered enterocytes at the apical pole have microvilli, the totality of which forms a brush border. On the surface of the microvilli there is a glycocalyx containing enzymes that ensure parietal digestion. Between the enterocytes there are goblet cells. These are single-celled glands that secrete mucus and endocrine cells. In the epithelium of the crypts, borderless enterocytes are found, and Paneth cells are located at the bottom. Borderless cells are cambial. Paneth cells, distinguished by their oxophilic granularity, produce a secretion that affects the process of protein breakdown, which is why many authors consider the crypts to be digestive glands. The lamina propria extends between crypts and into the cores of the villi. Solitary and aggregated lymphoid nodules are found in the lamina propria. They are more numerous distally in the intestine. The muscularis mucosae consists of 2 layers of muscle cells, the inner, circular, and the outer, longitudinal. The submucosa is well developed, composed of the connective tissue containing lymphoid nodules and blood vessels. The muscular layer is formed by 2 layers of smooth muscle cells. The serosa surrounds small intestine from all sides, except duodenum that is covered by peritoneum only in front. The structure of the different parts of the small intestine has its own features. In the duodenum, the villi are shorter and thicker, often irregular in shape. In its submucosa there are the terminal portions of the tubuloacinar duodenal glands. Their ducts open between the villi. In the jejunum and ileum, the villi and crypts are elongated and regular in shape. There are no glands in the submucosa. In the large intestine, intensive absorption of water occurs and feces are formed. The mucous membrane forms circular folds and crypts lined with simple columnar bordered epithelium. In contrast to the small intestine, there are no villi. The epithelial layer is represented by colonocytes, goblet, endocrine, and undifferentiated cells. The number of colonocytes and goblet cells is approximately the same. The muscularis mucosae is more developed and consists of 2 layers, internal, circular, and external, longitudinal. There are many lymphoid nodules located in the lamina propria and submucosa. The muscular layer consists of 2 layers of smooth muscle tissue. The outer layer is represented by 3 thick bands Taenia coli. On the outside there is a serosa. The posterior section of the digestive system is represented by the caudal part of the rectum, which ensures the excretion of feces. The anal canal is divided into 3 zones: colorectal, transitional, and squamous. The epithelium of the crypts of the colorectal zone is simple columnar. The anal transitional zone possesses a stratified cuboidal epithelium continuing into a stratified squamous non-keratinized, and then in stratified squamous keratinized one. In the muscular coat, smooth muscle tissue is replaced by striated skeletal tissue, forming the external sphincter. The serosa in the upper part of the rectum is replaced by the adventitia in the lower part. Dear audience, today we have thoroughly examined the features of the histological structure and functions of the main parts of the human digestive tract. In the upcoming lectures of the special histology course, we will continue studying the structure of other organ systems. Stay with us for our next meetings on the HistoNavigator channel.